Welcome to science class. Today we are going to learn about the structure of deoxyribonucleic acid, DNA. But before we do that, I want to give you the short background. The history of the discovery of DNA and its function is pretty extraordinary. For decades, we knew that organisms contain DNA, but we had no idea what its function was, what exactly it was made of, or what it looked like. Many of the mechanisms behind heredity had been uncovered in the late 19th century, and in the early 20th century, many scientists, such as Thomas Hunt Morgan, made additional progress. But still, nobody knew about the role of DNA. The term gene was coined in 1909 and was used by geneticists long before they knew anything about DNA or what genes literally were. In fact, Thomas Hunt Morgan said during his Nobel Prize acceptance speech that there is no consensus of opinion amongst geneticists as to what the genes are, whether they are real or purely fictitious. Was gene purely a metaphor, or was it a real physical thing? Nobody knew. Then in the 1920s, a British bacteriologist named Frederick Griffith discovered something really amazing. Griffith studied strains of Streptococcus bacteria, some of which cause pneumonia, others which don't, and they look different on a slide as well. When Griffith mixed one strain of Streptococcus bacteria with another, one strain would turn into the other. One species of bacteria transformed into a different species, like a cheetah turning into a lion. This is not heredity in the conventional sense, because bacteria do not sexually reproduce. Bacteria divide and make clones of themselves. But how could one species reproduce asexually and produce a new species of bacteria? Well, Griffith discovered a phenomenon known as horizontal gene transfer, but he didn't know it. In fact, Griffith's transformation experiment did not register that much attention in his lifetime. And then sadly, Griffith was killed during the Blitz, which was the Nazi bombing campaign of London during World War II. Much later, it was discovered that fragments of DNA called plasmids from one bacteria actually leave the bacteria, infect another, and then that bacteria now incorporates that gene or those genes from the plasmid into its genome. Bacteria don't have many distinguishing features. Their phenotypes are almost down to their chemistry, and bacteria are very simple. So this one small genetic change was enough to turn one species into another. In the 1940s and 50s, other scientists such as Oswald Avery, Alfred Hershey, and Martha Chase gradually discovered that the transformation that Griffith discovered is entirely dependent on the presence of nucleic acids. DNA was the molecule of heredity. This was particularly surprising because most scientists thought of nucleic acids as dumb molecules. Even scientists have blind spots. In fact, for decades, scientists had known that chromosomes exist, but they had no idea they were tied to genetics. Chromosomes, if you don't remember, are composed of histone proteins and nucleic acids bounded together. Proteins are so important in other life functions that it was assumed that some kind of protein was the stuff of heredity. But it turned out it was the nucleic acids within the chromosomes that contain the actual genes. That was not fully understood until the 1950s, practically yesterday in terms of history. Now there is much more to those stories, but I'm just using them as an introduction for this video. Once it was finally understood that DNA alone is the molecule of heredity, there was a massive rush to discover everything we could about the structure of DNA and how it worked, which is the real topic of this video. Let's get started. Many videos ago, I told you about the 4 basic macromolecules of life: carbohydrates, proteins, lipids, and nucleic acids. DNA is one of the 3 nucleic acids, along with RNA and ATP, the energy molecule. DNA is by far the largest macromolecule, and it's the second most common organic molecule in the body. DNA is made of just 5 different atoms: hydrogen, oxygen, nitrogen, carbon, and phosphorus. But those atoms don't just combine together to make DNA. DNA is made of subunits called nucleotides, which are repeating units which are themselves made of subunits. Yeah, I know, don't worry, I have pictures for you. A nucleotide will contain a 5-carbon sugar called deoxyribose, a phosphate group, and one of 4 different nitrogenous bases. Those nitrogenous bases are adenine, guanine, cytosine, and thymine. DNA is like a chain made of between several thousand or tens of millions of nucleotides, all strung together. In humans, our largest chromosome is made of 247 million nucleotides strung together. If those nucleotides were 1 millimeter in length, this one single chromosome could be stretched from my house to San Jose. String out all 46 chromosomes at this scale, and it would stretch all the way to Anchorage, Alaska. These nucleotides can be arranged in any sequence. There is no real pattern. Now that being said, you can pick any 2 humans and their nucleotide sequences will be more than 99% the same. All I'm saying is, in the big picture, a nucleotide with an adenine doesn't have to be followed by one with a guanine for any reason. Side note, it's pretty common to refer to the entire nucleotide with its sugar, phosphate, and nitrogenous base in terms of just its nitrogenous base. In books and magazines and other videos, they'll give you a string of DNA with nothing but the letters A, T, G, and C. It's just to make things more simple. Now, all of the information surrounding which atoms and simpler molecules make up DNA was known by the 1950s, but still, nobody knew what the structure of DNA was. This was crucial to know because in biochemistry, function follows form. Proteins look strange and random to us, but a protein has to look exactly the way it does in order for it to perform the task that it has. Knowing the structure of DNA would allow us to understand how it passes on genetic information, instead of just knowing that that's what DNA does. Today's world of genetic engineering and the near future of gene editing could never have occurred without this fundamental knowledge of DNA. So to sum up then, DNA at this point was like a giant Lego set. We knew which pieces to use, but we had no instructions, and there was no picture on the box to tell us what the finished product was supposed to look like in the end. The next big clue in discovering DNA's structure was made by Erwin Chargaff. Erwin found that in any sample of DNA, the amounts of adenine and thymine are practically identical, and the same held true for cytosine and guanine. For example, in the human genome, there's around 31% adenine and 30% thymine, and just under 20% for guanine and cytosine. Add all those numbers together and you get 100%. The proportions for each organism vary, but the pattern of percent adenine equals or nearly equals percent thymine, and same for guanine and cytosine, was always true. But it was anybody's guess as to how or if this pattern was evident in the structure of DNA. One of the last pioneering scientists who contributed to revealing DNA structure was Rosalind Franklin. Franklin was overlooked in her time, but is now happily celebrated, for she revealed the general shape of DNA. Using a technique called X-ray diffraction, Franklin bombarded samples of DNA with X-rays, which would reveal a pattern on the X-ray sensitive film. I don't know exactly how to interpret this photo, but smart people like Franklin understood that this pattern meant that DNA had a spiraling structure. Beyond that, though, she didn't know. Franklin was also a chemist, and a brilliant one. She figured out that the sugar and phosphate groups of DNA made up the backbone. They were on the outside of the molecule. And she did predict a double helix structure, ruling out the triple helix idea, but never put together an actual model. Piggybacking on all of this knowledge, James Watson, an American biologist, and Francis Crick, a British physicist, eventually created the first accurate model of DNA. They borrowed Franklin's research in a pretty unethical way to help them understand the spiral structure and the orientation of the model. And they used Chargaff's finding to realize that DNA is double-stranded, and in the middle, the nucleotide bases bond to each other according to Chargaff's rule. Where there is an adenine on one side, a thymine has to be found across from it, and the same is true for guanine and cytosine. Still, they never saw DNA. Nobody has. It's too small. but their model made all of the scientific predictions it needed to be considered the true model. DNA is made of what we call complementary strands of nucleotides. A nucleotide with an adenine is complementary to one with a thymine, so they always line up across from each other. But again, the order of nucleotides along the backbone of DNA can be anything. It's just that on the opposite side, the rule of base pairing, as it's called, has to be preserved. DNA is always organized with the phosphate and sugar groups in an alternating order along the backbone, and the nitrogenous base is always attached to the deoxyribose sugar. Each complementary strand of DNA is also upside down with respect to the other side. Even the spiral of DNA follows a rule. It is right-handed. This is called the right-hand rule, and you can see it with your right hand. Take your hand, curl your fingers. The direction your fingers curl, which is a counterclockwise direction, is the right-handed direction. DNA curls that way. All DNA in all organisms follows the right-hand rule, almost like we're related or something. All of these facts about the DNA structure reveal how it stores information. The base pairing rule and the non-random structure of DNA showed us that DNA stores within itself the information it needs to replicate itself. Imagine if we only had one half of the DNA helix. Well, we still know exactly what goes on the complementary side because of the rule of base pairing. This ensures that before a cell divides, it can accurately make a copy of the DNA. A lot of work was still needed to understand exactly how DNA is used to express the genes it contains, but this DNA model was the foundation to all of that and much more. That concludes this video on the structure of DNA. Next time, we're going to peer into the mechanism of DNA replication and see exactly how each of your cells ends up with a mostly perfect copy of DNA after mitosis. Thanks for watching.